Hello and welcome to my class. In this video, I'm going to talk about the digestive system of insects. I have divided this topic into two parts. In the first part, that is in this video, I'm going to talk about different food habits of insects and a general anatomy of their alimentary canal. In the second part, that is in the next video, I will talk about the process of digestion. So let's begin with the different food habits of insects. Here you can see an ant collecting food from the carcass of another insect. Here is a beetle who is feeding on the petals of a hibiscus bud. A bee collecting pollen and nectar from flower. And here is a mosquito who is feeding on blood. So these glimpses will tell you that different insects can have different kinds of food habits. Not only that, for the same insects, sometimes the younger form and the adult form can also have very different kind of food habit. If you think of a Lepidopteran caterpillar, you would know that they feed on solid plant material, whereas the adults do not even have the mouth parts to feed on solid material. They have siphoning mouth parts with the help of which they feed on nectar. This mosquito here, who is feeding on blood, in its larval stage, it was aquatic and it fed on planktons. So, not only different kinds of food habits are present in different insects, even in the same insect, the younger form and the adult form can also have different kinds of food habits. I also want to grab your attention to the fact that these two insects are feeding on solid food and these two insects are feeding on liquid food. These two insects are feeding on food of animal source and these two insects are feeding on food of plant origin. So sticking to this idea, let me show you this chart and discuss the examples. Many phytophagous insects like locusts, caterpillars, etc., they feed on plant tissue of solid nature. Then we have many insects which feed on plant liquids like nectar, as in adult moths, and then there are hemipteran bugs who pierce and suck the plant sap. We have many carnivorous insects like mantids and odonats who feed on solid animal tissue. They are predatory in nature, so they hunt on other insects and feed on the tissue. We have many parasites and pseudo-carnivores like flesh flies who feed on solid as well as liquid kind of food of animal origin. And then we have bloodsuckers like bedbugs and tsetse flies who feed on animal food but only of liquid nature. Then we have some insects like female adult mosquito who can feed on nectar as well as blood, that is liquid of plant origin as well as animal origin. We have the bees who feed on pollen as well as nectar, solid and liquid of plant origin. Then scavengers or detrivorous insects like black flies or caddis flies, they feed on solid food of both plant and animal origin. And then there are some true generalists or omnivorous insects like cockroaches, earwigs, etc., who can feed on both solid and liquid food of both plant and animal origin. So you see that there are various insects with different kinds of food habits. Now, obviously, to digest this many different kinds of food, they would require specific modifications in their alimentary canal. But before going into specific modifications, let's look into the generalized anatomy of the alimentary canal. The alimentary canal is usually a continuous tube that starts with the mouth, ends with the anus, and it has three functional regions: the foregut or stomodium, the midgut or mesenteron, and the hindgut or the proctodeum. In the foregut, food may be stored, filtered, or partially digested. The midgut is the primary site for digestion and absorption of food, and the hindgut is responsible for food absorption and feces formation. If you look at the origin, you would see that foregut and hindgut are ectodermal in origin, whereas the midgut is endodermal in origin. Let's look into how this happens. Here is an insect embryo that has the yolk at the center, and this yolk is lined with the endoderm, the mesoderm, and the ectoderm. Now, the endoderm lines the yolk and forms the midgut. But the foregut and the hindgut are formed by invaginations of the ectodermal layer. The mouth is formed here and the anus is formed here at the end, but the extensions meet the midgut, that which is endodermal in origin, and form finally a continuous tube. So we have the foregut and the hindgut, which are formed by the invaginations of the ectoderm, which finally meet the midgut, which is endodermal in origin. That way, the foregut and hindgut are ectodermal in origin and the midgut is endodermal in origin. Now let's look at different parts of the alimentary canal. As I said before, it can be divided into three functional regions: the foregut, midgut, and hindgut. The foregut has further four regions: the pharynx, esophagus, crop, and proventriculus. The midgut starts with the gastric ceca, and then it is a tubular structure. And the hindgut has three regions: the pylorus, the ileum, and the rectum. You can see this black line over here as well as here. These represent the cuticular lining of the foregut and the hindgut. Because foregut and hindgut are ectodermal in origin, they are lined with cuticles. The midgut, on the other hand, is endodermal in origin. It does not have a cuticular lining, but it has something else known as the peritrophic matrix. We will talk about the peritrophic matrix when we talk about the midgut. But let's start with the foregut. The cells of the foregut are usually flattened and undifferentiated because they are not generally involved in absorption or secretion. Okay, absorption as well as secretion of enzymes mainly take place in the midgut. Foregut's main function is supplying the food from the mouth to the midgut. So here you would find usually the flattened cells which are undifferentiated in nature. So there is no difference in the kinds of cells present in the foregut. The cuticular lining tends to be unsclerotized, consisting only of the endocuticle and the epicuticle. If you look at the insect integument, you would find three layers: the endocuticle, the procuticle, and the epicuticle. The procuticle is the part which contains the chitin, which again forms the sclerotization. Now, because the procuticle is absent here, the cuticular lining of the foregut is unsclerotized. But in many insects, there are sclerotized spines which are placed backward and they help in backward movement of the food. Now, let's start with the structure of the foregut. The foregut is preceded by the mouth and the preoral cavity. The preoral cavity is formed by the mouth parts like labrum, mandibles, maxillae, labium, and the hypopharynx. The other parts kind of envelop the hypopharyngeal region, and the hypopharynx differentiates the preoral cavity into the cybarium and the salivarium. So here is the region where the salivary glands would secrete their secretion. The salivary glands can either be tubular, that is tubule-like or tube-like, or acinar. Acinar means that they would look like a bunch of grapes. They contain mainly water and enzymes. There are two main types of enzymes which are found in salivary glands. One is amylase, which breaks down starch, and invertase, which breaks down sucrose. But in some carnivorous insects, protease enzyme is also found in the salivary gland enzymes. Now, let's look at the foregut in detail. Foregut has four regions: the pharynx, esophagus, crop, and the proventriculus. The midgut is known as the ventriculus, so the last part of the foregut is also known as the proventriculus. The pharynx is concerned with the ingestion and backwards passage of food and has well-developed musculature. So with the help of these muscles, they kind of push the food inside the esophagus. Esophagus is usually a simple tube which connects the pharynx with the crop. It is often poorly defined in hemimetabolous insects which have incomplete metamorphosis, but in many adult holometabolous insects like who have complete metamorphosis, it is long, slender tube running between the flight muscles and back to the abdomen. Now let's talk about the crop, which is a storage organ. It helps in storing the food before the food is passed on to the midgut. It is an extensible part in many cases. So it has many transverse as well as longitudinal folds, and it is folded into a smaller structure when there is no food inside. But as food enters, the folds become flattened and it permits a very large volume increase. But in cases like cockroaches, the crop holds air when there is no food. So there is no change in volume, and in those cases, the crop is not folded either. The proventriculus, or the last part of the foregut, is very variable in form. It often comprises a simple valve at the proximal end of the midgut, that is here, which projects into the midgut. These are also known as the stomodeal valve. With the help of these plates, these pads, and the stomodeal valves, the proventriculus controls the passage of food from crop to the midgut, as well as it restricts the food from a back reflux from midgut to crop. Now, crop is the main storage organ for many insects, but in some cases, the crop can also be modified. Like in cases of butterflies and flies, the crop is like a diverticula formed from the esophagus. So it does not precede the midgut and it does not follow the esophagus. It is just a diverticulum from the esophagus. Sometimes it can precede the midgut as well as can have a diverticulum. But in some insects like termites and scarab beetles, the storage organ is not in the crop but it is in the hindgut. These are known as the paunch in termites and the fermentation chamber in the scarab beetle. We will talk about this when we talk about the hindgut. But first, let's talk about the midgut or the ventriculus. This midgut often exhibits regional differentiation at either the anatomical level or at cellular level. Many insects have gastric ceca at the beginning of the midgut, and the number of the gastric ceca can be different in different insects. There are three main types of cells in the midgut. The first one is enterocyte, second one is enteroendocrine cell, and the third one is intestinal stem cells. The enteroendocrine cells secretes the hormones which will help in regulation of the function of the midgut. The midgut, as you know, is responsible for digestion as well as absorption of food. Now, when the digestive enzymes are secreted, how the muscles move, everything is regulated by the hormones which are secreted by the enteroendocrine cells. Next type we are going to talk about is the enterocyte cells. These are tall and columnar cells and their membrane on the luminal side, that is here is the gastric lumen, and in the luminal side the membrane of these columnar cells can be folded bearing microvilli. They increase the surface area for mediation of secretion of digestive enzyme as well as for the absorption of the nutrients. Here I want you to pay attention to these mitochondria here, to the rough endoplasmic reticulum as well as the Golgi bodies. Now, we know that these cells are responsible for both enzyme production and release because they have a lot of endoplasmic reticulum and Golgi bodies in them. This rough endoplasmic reticulum will help in translation of the enzymes, which are basically the proteins, and the Golgi bodies will help in packaging of these proteins. Now, when this enzyme will be secreted, that depends on when the food enters the body. In some insects, the enzymes are produced and secreted when the food enters the body, and in some insects, the enzymes are produced and stored into small vesicles as precursors or zymosins. And these membrane-bound vesicles are released at the lumen when the food enters the midgut. Now let's talk about the intestinal stem cells. We know that enterocyte cells are the main cells of the midgut epithelium because they help in secretion of the enzymes as well as absorption of the nutrients. But these cells would have a time limit or a lifespan. When those cells get destroyed or they die, then there are backup methods of producing the cells and replacing the enterocyte cells. That happens with the help of these intestinal stem cells, which are present in groups known as nidi. In some beetles, the nidi are restricted to crypts, and these crypts look like a papilla from the outside of the midgut. So here is the gut lumen. These cells, as and when necessary, will differentiate into enterocyte cells and they will pass on here and they will look like a papilla from the outside of the midgut. Now there is a very special cell known as the goblet cell, which is found only in Lepidopteran larvae. These cells help in alkalinization of the midgut. They have a potassium hydrogen antiporous system that is driven by an electrogenic VATPase. So these cells have their outer layer which gets invaginated inside and they form the folds. You can see many mitochondria in these folds as well as the basal plasma membrane also forms some folds here. Now these cells absorb the absorb the hydrogen ion and secrete potassium ion through an antiporous system, and that way it alkalinizes the gut lumen. Now the midgut cells are these main types of midgut cells we have seen, but there are some more cells of the midgut. The midgut cells in Ephemeroptera, Plecoptera and Trichoptera have a large central cavity, but we do not know about their functions yet. In Drosophila larva, the midgut growth is mediated by increase in the cell size of the midgut epithelial layer. But in Lepidopteran larvae, there is a burst of stem cell division just prior to each mold, and that way the replacement of the cells happen through cell division, and also through cell division the increase of the gut volume happens. The larval epithelial layer is replaced entirely at metamorphosis by pupal epithelium, and the pupal epithelium persists throughout the adult life. The midgut epithelium of the larval Lepidoptera undergoes apoptosis and autophagy. So recycling nutrients and the remnant of these is discarded as a meconium at adult eclosion. Meconium is basically a lump of all these things together, which is ejected out when the pupa becomes an adult. Now let's look at the intraluminal pH zones of Drosophila melanogaster. You can see that the lumen of the midgut can have different pH zones. The anterior midgut will have a neutral zone and the posterior midgut will also have a neutral zone. In the neutral zone, the pH is between 7 and 8. But in the anterior midgut, there is an acid zone where the pH is less than 3. And there is an alkaline zone in the posterior midgut which has about 10 or more pH. So these different zones help in efficient digestion of the food. Now we are going to talk about a very special structure in the midgut which is known as the peritrophic membrane. You would remember that the foregut and the hindgut are lined with cuticular lining, whereas the midgut is lined with the peritrophic membrane or peritrophic matrix or peritrophic envelope. This comprises a proteoglycan gel on a framework of chitin microfibrils. So this layer basically works as a barrier between the food and the midgut epithelial layer. It can be of two types. In the type 1, the peritrophic membrane is produced by the entire length of the midgut. Here, the peritrophic envelope is produced as different patches. It is not synchronized over the whole midgut, but it is made of patches that join together to form the entire peritrophic membrane. In type 2, the peritrophic membrane is produced by specialized tissue known as cardia, which are present in the anterior midgut. Here it starts to produce the peritrophic membrane, which then extends and envelops the whole midgut. Type 1 type of peritrophic membrane is found in Coleoptera, Blattodea, Ephemeroptera, Hymenoptera, Odonata, Orthoptera, Phasmida, larval Lepidoptera and adult Diptera. Type 2 is not as common. It is found in Dermoptera, Isoptera, some Lepidoptera and larval Diptera. The function of the peritrophic membrane is basically working as a barrier between the food and the midgut epithelial layer. As you know that the whole epithelial lining of the alimentary canal is a single line of cells. So if the raw food enters midgut, it can damage that cellular lining. So peritrophic membrane works as a barrier between the food's abrasive nature, pathogenicity or toxicity, and it helps in protecting the midgut epithelial layer. It also compartmentalizes the midgut lumen into two compartments, the ectoperitrophic space, which is in between the peritrophic membrane and the midgut cells, and there is an endoperitrophic space, which is inside the gut lumen. So in between the peritrophic membrane, we would find the endoperitrophic space, and outside the peritrophic membrane and in between the peritrophic membrane and the midgut epithelial layer is the ectoperitrophic space. This compartmentalization helps in enzyme activities as well as in generation of a counter current flow of fluids that increases the efficiency of absorption of nutrients. Now let's talk about the hindgut. The hindgut has three regions: the pylorus, the ileum and the rectum. The hindgut is also bound by the cuticular lining as the foregut, but here the intima or the cuticular lining is much thinner and it is more permeable to molecules. Let's talk about the pylorus. Pylorus is the site where the food from the midgut and the excretory waste of the Malpighian tubules will mix. You can see that insects do not have a separate excretory system. Here the Malpighian tubules open in the hindgut of the alimentary canal and the excretory waste is passed with the feces. So here the food from the midgut and the excretory product from the Malpighian tubule are mixed. Now, this pylorus also works as a sphincter. Okay, it can be bound with circular muscles and it can work as a sphincter so that food only passes from the midgut to the hindgut and it cannot go back to the midgut. Let's talk about the ileum. In most insects, the ileum is a narrow tube between the pylorus and the rectum. Sometimes the distal part of the ileum is recognizably different and it is known as the colon. The cell type in most cases is just one single cell type. It has extensive folding in the apical plasma membrane and it is with abundant closely associated mitochondria. The basal plasma membrane may also be folded, although the folds are less expensive than these folds which are present in the apical region. Why do they have so many folds and mitochondria? Because these are the cells which are responsible for the absorption of the leftover nutrients and liquid which are not absorbed in the midgut. There are some other functions of the ileum as well. In termites, the ileum forms a paunch, and in scarabid beetles, it forms a fermentation chamber. This paunch as well as the fermentation chamber is the house of the microbes which are symbiotic with the insects. So these symbiotic microorganisms stay in this special ileal region and they are often facilitated with cuticular linings which can be like elongated spines or bulbous lobes so that these microbes can hold on to those structures. Okay, so these microbes' main function is secreting enzymes which can help in further digestion of the food. Similar structures are also present on the ileal cuticle of cockroaches and crickets that have a bacterial flora. Let's now talk about the last part of the hindgut, which is the rectum. The rectum is usually an enlarged sac with thin epithelia, except for certain regions which are known as the rectal pads, where the epithelial cells are columnar. These rectal pads help in water absorption. So, the cuticular lining in these rectal pads are much thinner than the other places here. There are usually 6 rectal pads which are arranged radially around the rectum and they may extend longitudinally along the rectum. But in some cases, they can be also papilla-like or papilliform like in Diptera. Now let's talk about the other roles played by the hindgut. So mainly function of hindgut is feces formation and absorption of the leftover nutrients and water. But in some cases, the rectum works as the site for respiratory gas exchange. We know that in suborder Anisoptera under order Odonata, which includes the dragonflies, the respiration happens through rectal gills which are present in the branchial chamber formed in the rectum. The gills are repeatedly branching filaments with the terminal branches just 5 to 10 millimeter in diameter, and these are heavily invested with tracheoles. Let's take a section from this branchial chamber and look into it. Here you can see the section of the branchial chamber of the rectum. Here you see the 6 pairs of gills, and at the base of these gill lamellae there are chloride cells. These chloride cells help in absorption of chloride ions and inorganic ions from the water as it is pumped in and out of the rectum during respiration. So basically water comes through the anus into the branchial chamber where there are these gills present, and these gills will help in gaseous exchange from the water, and then that water will be pumped out. But not only the oxygen is taken in and carbon dioxide is given out in this branchial chamber, but chloride ions and other inorganic ions are also taken up from this water. And that happens with the help of these chloride cells. So we come to the end of the digestive system of insects. I have, just to recap, there are three functional regions: foregut, midgut, and hindgut. Foregut and hindgut are of ectodermal in origin. They are lined with cuticle. The midgut is endodermal in origin. It has a peritrophic matrix or peritrophic membrane that lines the cells. In foregut, the food is basically passed and it has spines to help in the backward movement of the food. Very little digestion can take place in the preoral cavity or in the foregut. The cuticular lining of the foregut is thicker and the cell types are similar because there is no digestion or no absorption in the foregut. The midgut is the main site for digestion as well as absorption of nutrients. So these cell linings are abundant with enterocyte cells which help in secretion of enzymes as well as absorption of nutrients. Most of the nutrients are absorbed in the midgut, but if it is not absorbed here, then it can be passed on to the hindgut, where the ileal cells and the rectal pads can absorb extra nutrients or extra water from the food. The rectum and the hindgut is also responsible for feces formation. In some insects, the rectal chamber can also form a branchial chamber which helps in respiratory gas exchange. And in some insects, the ileum can have the fermentation chamber or the punch which will help in holding the microbes that are symbiotic to the insects. So based on different kinds of food habits, there would be many different types of modifications in these different parts of the digestive system. But this is the generalized form, and hope you like this video. In the next video, I am going to discuss the process of digestion. So please come back for that.